The following is a recording of the third session on the Saturday of the BBS UK conference. Cilia and the Eye is led by Dr. Helen May-Simera.
Good. So I'm really excited to be back because obviously I wasn't here last year. I'm also excited to be able to stand here and actually show you pictures from my own lab and introduce to you some of the work that I'm hopefully going to be focusing on, and it kind of answers your question a little bit about growing a full eye. So let me very quickly start off with this slide because lots of people have been asking me where I've come from. So this time I wasn't in America. I came from Germany, as Phil said, where I am just starting my own small research group at the University of Mainz, which is very pretty. It's close to Frankfurt, it's very easy to get to, and it's an open invitation. If anyone ever wants to come and see the lab and see what we do in the research, everyone's always very welcome. So I'm going to start off. For many of you, this might, you already know this, but for the few people in the audience that are kind of new to this field, I'm going to just introduce what cilia are. And why is it that, Phil has often mentioned the word cilia and how it causes the syndrome. So as humans and all living cells, all animals, plants, we're all made out of cells. So lots and lots and lots of tiny cells all come to make all living organisms and things on the planet. Cells are beautiful, wonderful little machineries on their own. There's lots and lots and lots of different types of cells. So you have skin cells and bone cells and hair cells, and they all come in lots of different sizes and shapes. Now, some of these cells have tiny little hairs sticking out from their surface. And on this slide, what I've shown you on the right, there's a picture taken with a very, very fancy microscope of the surface of some of these cells. And on top of some of these cells, you have this little hair sticking out, which I sometimes describe as a candle on a birthday cake, or like a signaling antenna or a radio antenna on top of a car. And these are called cilia. Now, the next slide, don't get too scared because it looks a bit complicated, but it's not. There's two different types of cilia. You have motile cilia, and these cilia, they all work together and they move. It's kind of like a brush, a sweeping brush. So all the little bristles on the end of a brush, they move dirt or fluid along. So those are called motile cilia. Now there's another type of cilia called the primary cilium, and this normally cells only have one of these cilia, and they have a completely different role. They actually function a little bit like a radio antenna, and for many, many, many years people didn't think they were important, and it was actually through Phil's work back in the late 90s that they found out that actually this signaling antenna, this primary cilium, is really, 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 really important. Because what it does is it helps cells communicate with each other. So it helps cells send and also receive signals and talk to each other. So that's what this primary cilium does. So I mentioned that the body has all different types of cells, and many, many, many of those cells have these cilia. So on the next slide, I'm again showing some more pictures of different types of cells with all these cilia on. So you can see that they look quite different depending on what cell they're on. But I think you can appreciate that, they all look similar in the fact that they're these little hairs. So the top picture on the right are the cilia in your nose. And here I find it almost looks like a plate of spaghetti because they're so long that when you look at them, they're just all tangled up. In the brain, you also have cilia. And again, they help move fluid around. In your ear, you also have special kind of cilia. And the cilia that I'm interested in are the ones in the eye. And I'm going to talk about that in a little bit more detail now. So Phil already spoke a little bit about the eye. And obviously, the eye is a very, very, very unique structure and a very, very important organ for people that are affected with BBS. And one of that reasons is because the eye has many, many different types of cells that have cilia or are comprised of cilia. So here's a human eyeball, and there are lots of different parts of the eye. So you've got the lens, and then at the back you've got a structure called the retina. Now, I don't know how many of you remember the old style cameras, proper cameras where you had to put film in. So those cameras, you had to put the film in. So the way I like to describe the retina, it's like the film in that camera. It's at the very, very back. It's a very sensitive, light-sensitive film, and actually, when you see it, it's very thin and it's kind of see-through. And when you look at the retina itself and break that down, you again have different layers. And some of these layers have a very special cell called the photoreceptor cell. And these are the cells that Victor's targeting in his experiments where he's trying to inject the new genes. And these are the cells that they're trying to grow in the dish, these photoreceptor cells. And what's so special about these photoreceptor cells is that the whole outer side, so the outer portion of these photoreceptor cells are actually cilia. They're actually primary cilia. So if we look at them a bit more closely, here's a picture. Again, this is a real picture that's just been coloured, so it doesn't really look purple and green in real life, but you can see all these little projections are actually the outer segments of these photoreceptor cells, and they are primary cilia. And as Phil was mentioning, there's two types of these. There are ones that look like rods, and they just stick up. And there's ones that look like ice cream cones, but inverted, and those are called the cones. And these are actually modified primary cilia. So if these cilia aren't working optimally, so if they don't work very well, then the cell gets very unhappy and starts to break down. And so what happens to your whole retina, so that film in the camera, if the cells are there, then the film is very thick and it can work properly, it can work nicely, because you've got a nice retina. But if those cells aren't happy, then they'll start to disintegrate, which means that the retina gets thinner and thinner and thinner. It's almost like you're having holes in that camera, in the film, and that's how you start to have problems in your eyesight. Now, the way that BBS is involved in all of this is that the BBS genes tell the cells how to make different proteins. And I like to describe proteins like building blocks. You've got your Lego kit, and all the proteins have to come together, and they have to be put together in a certain way. And if you have lots of BBS, a lot of the BBS proteins, their job is to make and build that cilium. And so what happens normally in a healthy cell is that you have all these BBS proteins, they come together, they build this beautiful cilium. And if you have a problem and a mutation and a mistake in one of these proteins, for example, if one of those Lego blocks isn't fitting together, or if maybe it's got a corner or a chip broken off of it, then the cilium can't function optimally. It's still there, it's just not able to do its job. It's kind of, imagine if you've got a car, you've still got the car, but if the radiator's broken, it's not going to work as optimally. And so obviously, the more often that you've got problems with the radiator, then, the car's not going to be very happy, and it's most probably not going to drive that far. So that's generally what we've always been interested in and what we've been looking at. I've been very, very, very lucky because I was able to get my own grant to start my own lab. So I'm going to show you some pictures of my own lab at the University of Mainz at the end, and I'll explain a little bit about exactly what it is that we particularly are doing in that lab. So this is my team, this is my lab. We're actually in the basement because I think that's where everyone starts out. And I'm very, very, very fortunate because I've got some fantastic people that help me. So this is my lab manager, and this is my scientist. And I've also got students who I'm very excited because they're starting to get excited about Cilia too. And this one student, actually this one, she's decided to come and stick around and stay with us and actually do her master's project with us. So that was exciting. I've also got some fancy equipment, and I was very excited to actually be able to buy my own stuff, so we can, do a lot of the analyses and look at the different cilia in the different cells. This is another, the actual scientist that does a lot of the work in my lab. And what we're actually doing is that we are actually looking at how this cilium works. So how does it work as this signaling antenna? How do the cells communicate? How are the messages sent to the eye or from the eye? And what can we do to help the cells that are unhappy because their cilia aren't functioning? Because you can't just take the cilium off and put a new one on. That's going to be difficult to do. But we're trying to understand is what this signaling antenna is actually telling the cell to do, so that we can then possibly replace whatever is missing and whatever signals are missing. And most people in the scientific community are working on these photoreceptors, and they're focusing on the rods and the cones. However, there's lots of other cell types in the eye, and there's actually one very important cell type that, there's one word that I want you to remember from my talk. It's the RPE. So the RPE is actually a cell layer behind the photoreceptor, so it's behind the retina. And these cells are the caretakers of the photoreceptor cells. And all this work is going on to generate, maintain, build new photoreceptor cells. But no one really is focusing on the fact that these RPE cells also have cilia. And these cells are important for nurturing and looking after the photoreceptor cells. And that's where I come in. That's why I thought we need to also look at these other cells. Because if we know that those cells are ciliated and they're not looking very well and they're not happy, we want to make them happy. Because if those cells are happy, then they can support the very, very sensitive photoreceptors. So that's what we're doing. So we're trying to improve and help the growth of the RPE cells, which will then support the photoreceptor cells. And so the picture I have here is one of my favorite pictures, because you can see this big square that looks like it's got kind of a mosaic of green tiles, or black tiles with green borders. And those are actually the junctions between the RPE cells in the back of an eye. And we can see that every single cell has got one dot. The dot is actually a cilium because it's a view from the top. And that was, for a long time, people didn't really know that these cells had cilia. So it's quite nice to be able to work on something that other people have been ignoring. And we hope that, the more we learn about it, the more we learn about how these cells function, the more that we can support photoreceptor cells, which are the ones that start to become unhealthy in the patients that are suffering. And with that, I want to extend an open invitation, as I mentioned before. I really, really would love to see anybody, if you happen to be visiting Mainz. The other thing I really wanted to mention, I was just thinking about this when I was sitting listening to Phil, is that, so I've been in Germany for about a year now, and last summer I was invited to go to the German Patient Society, which is much, much smaller. I mean, there was only about 20 families, much, much smaller. They only meet every 2 years, and they were very, very, very keen to learn about the clinics, because they really want to implement that as well. But not having the NHS means that it's much, much more difficult to coordinate. And I just realized just how special and lucky you are to have the NHS that allows you to do this. And I know everyone always complains about the NHS, but it is brilliant, and you're so lucky. So with that, my last slide, I know it's an old one, I know, but I love this slide. And I know there have been new ones, but for some reason, this one always captures my imagination. So I really, really, really want to thank the BBS community, because you guys are the ones that really give us the motivation, give us the drive, give us the reason to get out of bed in the morning, and do what we do. So with that, I take any questions. So that's very, very, very recently we've started looking at that. that yes, there are these things called exosomes and ectosomes, and we've been doing some preliminary studies, actually that student that I talked about, to show that cilia are not only receiving but also sending signals, which is obviously a complete game changer. Because we used to think they were only receiving, but now we know that might also be sending, but we don't know what they're sending, we just know that there are things coming off, so we think they are sending signals as well. I mean, we definitely know they're receiving, we know that. But we now know that they might also be sending signals as well. But what those signals are, where they're going, is something we don't know yet. But I think, so part of the reasons I was talking to you about these two types of cells, the RPE and the retina, that there's obviously some information exchange between these two. So that's something that we were looking at. So. I don't know if we've got time, but one quick question.
Is this work just related to BBS, or is it also related to eye conditions?
Absolutely. I mean, so my lab always uses BBS as a model, and because I have a lot of the animal models for BBS, but the information what we're learning is relevant to other ciliopathies and other retinal degenerations, especially in the eye, because a lot of diseases that aren't syndromic also result in retinal degeneration, and we think that the factors and the causes are very much similar and linked.